Thank you, Pims, for the very kind introduction. And it's really a great honor to be part of this today's very exciting webinar. I really learned a lot from Trevor's talk. And today I would like to really share some of our recent work and ongoing work on something we actually identify by surprise, a subpopulation which look like dormant in the tumor. And also that has some spatial pattern in the tumor distribution. So basically, my lab is interested in using, on the one hand, mouse model, and now increasingly using human brain organoid, of course, to study, on the one hand, neurodevelopment, neurogenesis, and neurostem cell differentiation. But on the same, in parallel, we also model brain tumors in mouse and now in human brain organoid model to study the similar processes, or the oncogenic processes in these models. And quite often they are linked because also, as you can see from the last talk, that many genes are actually important for regulating development and even injury response are also implemented in cancer. So it's obvious that I think it's interesting to study both, and then one can learn from each other. And scientists from Toronto really deserve the credit, I think, for discovering cancer stem cells from John Dick on leukemia. That was basically found by single cell can initiate the entire leukemia in immunocompromised mice, and similarly by Peter Dux, mentioned by the previous speaker, identifying the similar cell type in human brain tumors. And it's only subpopulation of that cell in brain tumor can do it. But here I try to, give a summary about heterogeneity, as we already heard that on the genetic level, there's different clones, like maybe carrying different mutation, copy number variations in the same tumor, and they are of course very different if you analyze their profile. But also we know that within the same clone, there are also stem cell hierarchy, like in a normal tissue that's mostly share the same genome, but you have stem cell differentiation hierarchy observed during development, and similarly, similar things have been found in cancers. So that means also we have these two types of heterogeneity. But on top of that, that can be niche difference, vascular niche, hypoxic niche, and I think all this probably played an important role in influencing heterogeneity and also, of course, influencing the therapeutic outcome. I think it's very important for us to really dissect these things one after another, really to get a complete understanding of this. And on top of this cellular part, there is also very clear metabolic heterogeneity in the tumor, and as I think is one of the hallmark of cancer metabolism is that really activation of the glycolysis pathway, as postulated initially by Otto Warburg really some time ago. And actually, Otto Warburg was a MD student in Heidelberg, where actually he did not discover the glycolysis pathway. That was mostly defined by his friend Otto Mayerhoff in Heidelberg at the time in the Max Planck Institute. He's the guy, and this glycolysis pathway was also called Embden-Mayerhoff pathway because he's the one really identified most of the essential regulators in glycolysis pathway. And his interest on metabolism was basically initiated actually by Otto Warburg when they met in Heidelberg. And the idea is that cancer cells turn to utilize glycolysis pathway. I think there are different hypotheses why they do that, but I think this has been observed in cancer in general, general phenomenon. And actually, Heidelberg was a center for metabolism. Even the ATP was discovered in Mayerhoff's department by Karl Lahmann, who is standing here. So it was a very interesting time, I think, at the time. So our interest in brain tumor developed from studying brain development at the time when I was a postdoc. Later also in my own lab, we studied this gene called TALIS, or it's one of the nuclear receptor gene. We also call it NR2E1. And it's one of the Heidelberg genes, actually, because that was when Christina Faulhaber was doing the Heidelberg screen to study mutants in Drosophila, like to discover gene which regulate Drosophila development, and TALIS was one of them. You can see the name. Actually, it's both Hetelis and TALIS in the TALIS mutant fly. And later it was found that actually Taylor's this gene in mammals is only expressed in the brain, but we have seen that in the subventricular zone, the so-called adult neural stem cell area, Taylor's, and also in the subgranular zone in hippocampus, actually, Taylor's is expressed by stem cells in this area, not by progenitor cells. And if you remove this gene, you lose stem cells in the adult brain. And if you give 2 more copies of this mouse gene into these cells, and these cells get much more, they get more neural stem cells and also brain tumor formation from these animals. So that was, I think, provide a very interesting link between stem cell expansion and brain tumor formation in vivo, because the manipulation was really mostly done, only done in stem cells. In addition, if the tumor is fully developed, we also have seen that Taylor's is expressed by the stem cells in the tumor, and if you remove Taylor's gene in the fully established tumor, genetically, the animal do survive longer, suggesting actually as a self-renew gene. So if you really target this pathway, the animal do survive better. So yeah, like as a proof of concept, if you target cancer stem cell, that might be helpful for therapeutics. And then we further decide to really use this gene as a reporter to study, how tumor develops and to really get to understand the tumor heterogeneity in a general view. I just show you a few slides on this. So the mouse model we use here is the nestin-TV model pioneered by Eric Holland. And the oncogene we use the ARCase to deliver is the PDL-B plus AKT. Basically, you activate receptor tyrosine kinase and PS3 kinase pathway, and almost all animals get tumors. You can see here there's classic necrotic area and also angiogenesis that animals die very quickly. They are very proliferative tumors. So this is a very efficient model to, we can build on top of that further genetic tricks. And you can, of course, implement the luciferase gene in this model and to see basically how the tumor developed. Here we image like 23 animals with luciferase, as like basically 3 days interval. And we basically give this data to a modeler. Lisa, she's a mathematician. Then basically when she was trying to plot the data showing that basically the tumor in this model actually grow exponentially because that's most of the model, tumor growth data fit to the exponential model. And it's and you can also calculate doubling time of this model, of the tumor growth. It's a very aggressive tumor. But actually, if you consider the stem cell, non-stem cell compartment for the tumor to grow exponentially, it's actually quite difficult. And she was trying to fit that in a modeling way, that the only scenario she found that the tumor can grow exponentially is to let the stem cell migrate toward outside. So that's the only way the tumor growth can fit to what we have seen. But that's very easy. All other ways, basically, you cannot achieve, I think, a very optimal exponential growth from her testing. And additionally, she found that if you allow every cell to migrate in this model, green indicate like the stem cell, red as a non-stem cell, basically you get a sphere-like structure. And if you allow stem cell to migrate, you get a branching, very invasive phenotype, which is, I think, fit. Basically, if you section this model, you will see the green stem cells stay outside. And that's, I don't think for this audience I need to introduce that brain tumor, GBM probably is one of the most invasive solid tumor, and you quite often have this branching butterfly phenotype. And experimentally, it can be easily tested. We also have this Taylor's GFP transgenic mouse carrying the tumor in the TVA background. We can check that where are these cells at the beginning of tumor induction. As you can see here, the GFP is really mostly staying at the border of tumor during the tumor development. But this can also be like, there are also endogenous neural stem cells. We cannot distinguish the two. And we also use the transplantation sorting of the GFP tumor and transplant them, and then into a compromised mice. There's no Taylor GFP, of course. And you see this is a really large tumor, multifocal tumor. As you can see here, these are proliferating cells in the tumor, right? It's very large. And if you look at the Taylor GFP stand in brown here, although in the middle you will still see some cells, but actually they are mostly at the outside, at the border of the tumor. It's already everywhere. So I think that's additionally confirming that in this model, the stem cell also from beginning and to the end, they preferentially distribute at the tumor border. So from this we can understand that now the tumor grow exponentially. That's very interesting to know, and this pattern is driven by migrating kind of stem cells at the tumor border. And we also did a single cell lineage tracing in this model. I won't really introduce too much detail there. Then you can use that to build a model of the different cellular behavior. But then you can apply temozolomide to kill these fast dividing cells in the tumor and see what happened. From the model prediction, we can see that if you give temozolomide, the tumor will shrink very rapidly, but it will grow back very rapidly. Okay, so this is on the modular level. These are the real data. I show you 3 animals. So this tumor grows, then you apply temozolomide. This green time period, we apply the temozolomide. This tumor respond very quickly, as you can see here. Immediately they respond to the tumor to the treatment, and but they grow back very quickly. This has happened to all the tumor we have tested so far. And that means, of course, you don't really kill the stem cells with temozolomide treatment. That's why you expect this pattern. And if you block stem cells by in this model, you will see actually because stem cells in this case are mostly less proliferative compared to the manager block, and we won't see a dramatic response at the beginning, but in the long term, you will see a decline of the tumor growth. This we used manipulation of tailors because we know that if you remove tailors in this model, the tumor really do shrink a lot, and then they lose stem cells. Here we basically use the inducible knockdown system to let the tumor grow, and then we start tamoxifen treatment to induce tailors knockdown in this tumor and see what happens. Basically, that's what you see. So if you block tailors during the tumor development, so the tumor will continue to grow because tailor expression actually is mostly restricted to the slow dividing. Some of them are dividing, but mostly slow dividing the population. The tumor will continue to grow because the temozolomide responding cells are still there. But in the long term, it looks like these cells cannot, sustain themselves. They are known that to be progenitor cannot self-renew, and in the long term, you will see tumor signal drop. This is a very different pattern from the temozolomide treatment. And also, again, suggesting, I think, it's really essential to target cancer stem cells. We also did RNA-seq in this mouse model because we want to get the pure really population here using the Taylor's GFP, but we also label the tumor cell as RP. Then we can store the GFP RP as tumor stem cells, and GFP negative, RP positive as differentiated tumor cell, and then we can really sequence that in the pure population, see what's the difference between the stem cell and non-stem cell here, as indicated. DS red cell will be a non-stem cell, and you would see, of course, many stem cell genes are enriched in this GFP population. As also Taylor's and nestin, Has5. They are very known stem cell markers. And you will see actually classic neurodifferentiation genes like double coating, MAP2, many genes, OLIG1, and they are enriched in the differentiation compartment. So it's from the same tumor in the pure population. If you really sort them, you see clearly the molecular hierarchy is there in this on the stem cell differentiation hierarchy. But additionally, what is very interesting in this stem cell population is they activate many stress-resistant pathways. So the proteasome pathway are dramatically upregulated, heat shock pathways, and I think that's indicating probably there is a classic stress maybe related to integrated stress response. And so from this data, I think we also analyzed human single cell data. It looks really similar. I think also what Trevor showed is very clear, and from also many other single cell studies indicating both mouse and human GBM cells can be hierarchically organized into stem cell differentiation model on the molecular level. On the cellular, of course, yes, same. So, but how can we really target these cells? To really look for suitable target, Patricia, a talented postdoc who really decided to do this experiment using, again, the mouse model, and generating a tumor, and then isolating tumor stem cells, but also we isolate neural stem cell from mouse subventricular zone where we, derive the tumor. Then we performed ribosome profiling assay. The idea is that we pull down the RNAs which are being translated in the cancer stem cell and compare that with neural stem cell to look for, whether we can enrich anything, again, anything enriched particularly in the cancer stem cells. Of course, you will see many genes. There's a clear difference between tumor stem cells and neural stem cells. But then we decide our criteria of selection of target is that the target should be druggable, has to be enzyme, or shouldn't be a transcription factor so far because it's difficult to drug them. And better knockout has no severe phenotype. Suggests, we want to see that if we can develop a drug, maybe there's less toxicity. And the best would be should be only expressed in cancer stem cell, but not in neural stem cells. Then we can really study what is particularly associated with cancer stemness, which is actually little is known, because if you look at the stem cell genes, they are actually shared stem cell genes between cancer stem cells and neural stem cells, similar like Taylor's. So among these, targets, GPT-1 stand out very easily from our selection, and further confirm that by Western blot, because that's what we look for, translated RNA. So that should really indicate, we also confirm that with independent brain tumor stem cell lines compared with neural stem cell brain, really GPT-1 stand out. On the protein level is highly expressed in brain tumor stem cells. If you stain them in cultured neural stem cell, you really don't see signal, and you see the expression in cultured brain tumor stem cells as well. So the screen really works. We do find proteins are expressed in brain tumor stem cells. And if you look at in vivo, this is the mouse tumor induced in the nestin TVA model. Here, red stains GPT-1, green stain as PCNA, the proliferating tumor bulk, as you can also see here. This is in the subventricular, as you can see. It's actually interesting that, again, we see this pattern. The red cells, GPT-1 cells, are mostly distributed at the tumor border, similar to the Taylor's expression. So this, I think, reassuring that this pattern general correlation do exist, although you still see some cells in the bulk, but is very clear. I think that regulatory enrichment is at the border. And if you look closely, we will come back to that later. Actually, most of the GPT-1 cells are negative for PCNA in vivo. And if you look at normal mouse brain, it's a sagittal section here. In the subventricular zone, where we know the stem cells are stand with PCNA, I show you, the GPT-1 is not expressed. And we also know that GPT-1 knockout has no clear phenotype. And the only cells which was reported before express GPT-1 in the mouse brain is the cerebellar Bergman-Glia cell. There we see the signal. Okay, so this is the only cell express GPT-1 in the mouse brain. In a reliable manner, we can detect that. In human tumor, this is basically where we select the tumor border again, as you can see here, verified by new staining. So here would be the normal part of the human brain tumor, glioblastoma, and this is the tumor bulk. As you can see, that's where we select this area. There is where you see the GPT-1 staining in brown. So it's really, I think the pattern is similar from mouse to human. This is not only tumor we have seen. This in general, it looks similar. And we also have this tailored GFP tumor, right? So, do they co-localize? Yes, some of the cells, GPT-1 cells express tailored GFP, and not all of them are overlapping. So that's indicating there are some differences. But of course, here we are using the GFP reporter. Here we are using the protein staining. So that might be it's important to keep that in mind. And what is GPT-1? GPT-1 is the actually borrowing enzyme. It's Glycerol-3-Phosphate Dehydrogenase 1. Enzymatically, it's catalyzed conversion from the hydroxyacetone phosphate into glycerol-3-phosphate. And the reaction is reversible. And at the same time, during the reaction, it's generating NAD+. Of course, the druggable enzyme is an enzyme and regulating of clearly lipid metabolism. And this well studied in the East is responding to osmotic stress. It's basically used as the osmotic stress reporter. And little is known in mammalian cells, maybe because the knockout has no clear phenotype. And but if you look at the GBM TCGA data, the high expression patient do survive worse than a low patient, GPT-1 low patient. Now the same with the progression-free survival patient. So indicating that probably there is something interesting behind this. But if you look closely to the tumor, this is again mouse tumor. We can trace the proliferating cells with BRDU in the mouse, and you will see this is again the tumor box and the GPT-1 distribution at the border. But we don't really see BRDU positive cells are positive for GPT-1. We use different markers, Ki67 here, MCM2, another marker showing the same, and PCNA is similar. So these cells, although it express stem cell marker like TGP, although we got a screen by isolating the stem cells, get this target, but these cells are not proliferating in vivo, at the time we examine them. So it looks like they are dormant. And the same in human tumor. This is the tumor we got from Krista Hermendo here. You see the tumor box stand with Ki67, and these cells are not proliferating in vivo. And again, they are at the tumor border. So, and this sleeping cancer cell actually was a long time ago, was already realized, even mentioned by Otto Warburg in his very well-cited review. If here I quote his a sentence: the sleeping cancer cell will possibly play a role in chemotherapy, but from energy consideration, I could think this sleeping cancer cell could be killed more readily than growing cancer cells in the body, because if they're sleeping, probably they consume less energy from this point of view. But is that really true? So again, we apply temozolomide to this mouse model I described you. You see a clear difference, right? The response, then the tumor will shrink and they will go back. And then we decide to use this different stage to really analyze GPT-1 cells in this model, then co-standing GPT-1 with proliferation marker PCNA. This DMSO control, and showing that these don't really co-label, right? And that's basically during temozolomide treatment, the tumor is shrinking, so we also don't see these cells are dividing. And it's only when the tumor is relapsing, so you start to see these cells express PCNA, we basically confirm the same thing with the BRDO experiment during the same treatment. And once it's it's really reached the level of the regrowth plateau, again we see the original pattern. These cells start to dominate again. This is a quantification. So during normal normal developed tumor, you rarely see any co-labeled cells between GPT-1 and PCNA, and similarly BRDU. But during the relapse stage, many of these cells are proliferating. So it's really, I think, so temozolomide doesn't kill these cells, and they are somehow weak during tumor relapse. And it's not on a cellular level, but also on the RNA level. In the same model, we performed the RNA-seq in the independent experiment. Basically, wait the tumor, wait until the tumor develops. We apply temozolomide, and 2 weeks after, we collect tissue for RNA-seq. This entire tumor tissue. And there we had even anti-VEGF treatment for the mouse. Basically, here is the control. VEGF really does nothing from the RNA-seq level. You will see, but when you apply temozolomide or temozolomide plus VEGF, basically you see the tumor, proliferating cells indicated by PCNA or marker like Ki67, they are much less from an RNA data, but GPT-1 get even enriched. So you really don't touch these cells by applying temozolomide. But if you now really inhibit GPT-1 using the similar trick I showed you for the tailless inhibition, again, inducible knockdown based on CRE-ERT2 system, that basically we wait for the tumor develop animal to develop tumor, then apply tamoxifen. GPT-1 is knocked down. In this case, the animal do survive longer. So GPT-1 is solid, let's say, validated target to block stem cell and achieve animal longer survival in this model. What happened to these cells? So there we decided to use CRISPR to knock out GPT-1 in a cancer stem cell, perform the RNA-seq. Then again, you will see basically many of the differentiation genes are upregulated. as we have shown that H7 is important for differentiation and double counting. Many of the genes was found in the tail GMP negative RNA sec, and they are upregulated in the GBD1 knockout with 2 different guide RNAs showing the same thing. And many of the stem cell genes, also proliferation genes, are downregulated, like nestin and CD44. Even Taylor's is downregulated upon GBD1 knockout. So the cells actually tend to differentiate as well because they tend to attach in culture. And also, surprisingly, this is something we are still working on, that we do see MYC target are dramatically downregulated. It's almost black white, and also E2F target. So suggesting maybe we are also inhibiting protein translation there. That is interesting because we do see the proteasome stress genes are regulated in the cancer stem cell by tailored GMP RNA sec, right? So I think this is echoing that finding that is something very much related. We also did the lipidomic assay, basically seeing that actually neural stem cells are very different from tumor stem cells because GBD1 is regulating lipid metabolism. And if you knock out GBD1, basically you reshape this lipidomics. And one of the most differentially affected pathway is basically the glycerol-3-Phosphate metabolism as expected. And I show some example of this lipid species that is upregulated in brain tumor stem cell compared to neural stem cell. But if you knock out GBD1, many of this can be reversed back to normal stage compared to somehow we can reverse toward neural stem cell metabolism, indicating probably GBD1 is somehow driving the cell toward cancer stem cell phenotype on a metabolic level. And in human cells, here it's similar. We use 2 independent patient-derived cancer stem cell line from Christopher Menzel, use basically 4 different siRNAs against GBD1, perform independent limiting dilution assay. The more flattened the curve is, the spheres you will see from this assay, indicating that from both of the cells, we've confirmed that GBD1 is required for the tumor stem cell to maintain themselves in the sphere assay. So it's basically similar to the mouse study. And interestingly, if you look at the RNA expression of GBD1 among the other cancer, this what we plotted from the TCGA data, I think. Endogenously, GBD1, because it's regulating lipid metabolism and osmotic stress, is found in kidney and in liver. That's where you see also tumor cells do express that. But the next way is really GBM has the highest level. So it looks like there's something in GBM probably specific to activate GBD1 expression. So basically, what GBD1 does, what all this finding suggests that also together with the first part of what I showed you, that the stem cells, relatively speaking, they are more enriched at the tumor border. And I think that makes sense because the tumor in the middle is very hypoxic and less proliferative, and there's a lot of necrosis going on. But these are the temozolomide resistance, and they clearly drive cancer relapse because they start to divide after chemotherapy. And this, because they're at the border, I think they are very infiltrative, and they are stress resistant because I think PD1 is basically classified as a stress responsive gene. I think that's indicating that this is basically probably a very important regulator for stress resistance in cancer stem cells, and also indicating that the lipid metabolism is very important in regulating cancer stem cell behavior. And our working model so far is that I will try to elaborate what does mean, and we are still trying to put that into context. That this is basically glycolysis pathway, right? So and you generate lactate, and these cells, because I think they are fast dividing cells, they require the product from the glycolysis. That's what most people suggest. But what we see here is a very distinct pattern that the GBD1 is really mediating this branch of metabolism. That GBD1 is upregulated in the subpopulation of tumor cells, and this may drive the energy flow toward, or dilute energy flow toward the lipid or glycerol, basically, metabolism. And these cells are probably less glycolysis, and they store the energy, and they are dormant. At the same time, also they generate 88+, to make them more resistant to stress, I guess. And But this entire thing can be reversed because it looks like, it's hibernation. You store something in the winter and you can come back after winter. And after chemotherapy, this cell can reverse back and merely, I think, making use of the energy they store. That's why probably if we block GBD1, this cell cannot reverse it back. That's the idea. And because the reaction GBD1 is mediating is reversible, right? And interestingly, there's something I think linking GBD1 to POC pathway because Sabatini's lab recently found that along this glycolysis pathway, let's say, they test all these different metabolite. Is only the DHAP is sensing or is giving the signal to TORC activation in this pathway, knowing that mTOR do respond to glucose concentration, right? And I think there's something going on. GBD1, we also see that in the GBD1 knockout cells, TOC pathway activity is basically completely blocked in this model. And that's may linking to the protein translation effect we have seen. So I think there's probably more study need to be done on this one. And it's not only something really specific for glioblastoma, and nature has actually used that in many different ways. The more I'm interested, the more interesting thing you will see. This is like a study really some time ago, found that in a Chinese silkworm, they also, the embryo, if they're born at different bad time during the year, they will undergo diapause, waiting for the right season to further develop. And during diapause, basically, it's like hibernating there for this animal, and they basically store glycerol. And when they exit diapause, this glycerol is gone. And of course, hibernating black bear, if you measure their metabolism, the only active one is glycerol metabolism during hibernation. It's not really glycolysis. And Glycerol-3-Phosphate is also used as a systematic immunity, mobile inducer of systematic immunity in plant. And in this very interesting sea algae that can grow in very high salt concentration, and they did that by inventing this evolving into a new enzyme, basically by combining the GBD1 activity, DHAP to G3P, combining the enzyme between, this is like this GBD1 version. So this enzyme is basically fused into one. So then you have that 1-in-2 conversion. This green algae can convert DHAP into glycerol directly in one reaction, and that's why they can grow in high salt seawater. And also in pancreatic cancer, so the Genentech developed a LDH inhibitor. There they have seen that if you treat these cells develop resistance, but one of the major metabolites are regulated, and the resistant cell, as you can see here, is G3P. I think looks like there is some metabolic flow or plasticity. If you block glycolysis pathway, they also activate this pathway for resistance. And so on. There is a interesting fish growing in a freezing condition, but these fish actually, what they do is how they can manage that is by activating GBD1 expression during winter. Before winter comes, they activate GBD1 expression. They can store glycerol, and this help them to grow or swim in freezing condition because it's protecting them from this freezing condition. In GBM, there was one study I found that's quite interesting, that they used different PET tracer to show or compare the different PET tracers. This is like a glucose tracer. People know that they are not really specific for tumor. And this is the choline tracer, which we know that this really traces lipid. And we know that if you want to make the choline tracer working, is require glycerol-3-phosphate, because there's a lot, I think there's a Kennedy reaction that requires the glycerol-phospholipid to make this work. And that we see very nice, I think less background, but very sharp imaging. And actually, this PET tracer also can show very nicely the tumor border, at least from this study, what I can see. It's actually nicely fitting to what we see. This enzyme is more enriched than the tumor border. So this is, we think how it is working, that the cancer stem cells are under huge stress because the tumor environment is really very stressful. There's a massive cell death going on, and somehow the stem cells manage to activate the program to protect them, or basically hijacking the normal stress-resistant pathway and they also phenotypically undergo a dormant transition. And that give us really interesting, therapeutic target possibility, because you can develop drug to target this enzyme. But we also know that normal cells really don't use that mechanism because they are somehow cancer specific. This may give us a therapeutic window of treating these cells and avoiding the normal cells. So what is ongoing at the moment, we are trying to understand how is GBD1 regulated. I think we identify what is really triggering the GBD1 expression in brain tumor stem cell, which I don't have to show you. And it's very much linking to actually current treatment of glioblastoma. And we are collaborating with a pharmaceutical company to develop GBD1 inhibitor. Now we are already testing them in an organelle model we have. And also we are using GBD1 as a reporter to really study dormancy and metabolic heterogeneity. With that, I would like to finish by thanking our collaborators. On the modeling part, we collaborated with Thomas Hiffer and Lisa. And the human cell knockdown was done by Yunhe with Bernard Radwimmer. And also we got cells from Peter Engel. And the screening was done by collaborating with Bernard Buchhaus' lab. And the metabolomic assay was done in EMBL. We got a lot of sample and collaboration from Christoph Hermann in Heidelberg. And this is our funding agency. Thank you. So I'll be happy if there are some questions.